Alright guys, welcome back to Underground Science, and in this video we'll be talking about the byproducts that are produced in the Krebs cycle. Alright, so things like NADH, FADH2, things like substrate level phosphorylation, and other things. Alright, so let's go and get started. So I didn't draw the mitochondrial cell here, but just know that everything we're going to be talking about is going to be happening inside the mitochondrial matrix. Alright, so everything that we're going to be talking about here is going to be happening in the mitochondrial matrix. Alright, and let me just state that one of the enzymes, however, are going to be bound. So let's see how to, one of the enzymes, I was going to say one of the enzymes are going to be actually, it's not going to be completely in the mitochondrial matrix, but it's going to be bound also on the cristae. So one of the enzymes, however, will be bound on the inner mitochondrial membrane, alright, or also called the cristae of the mitochondria. So let's go and get started. So if you haven't watched my previous two videos about the Krebs cycle, the first video I made about the compounds that are actually produced in the Krebs cycle, then the next video I made a video about, or in the next video I talked about the the enzymes involved in the Krebs cycle, right? And so that covered everything except for the byproducts produced, like in some of the reactions, all right, like NADH, ATP, and things like that. So that's what we're going to talk about in this video. So if you don't see some things like mentioned, like small things like water being added or CO2 being added, just know that we talked about it in our previous videos. So make sure to go check those videos out. All right, so let's go and get started. I said that a lot now, so let's go and actually get started. So let's start with our first molecule produced in the Krebs cycle, which is citrate. All right, and we'll work our way back or work our way forward. So first, citrate, remember, is converted to isocitrate, and remember this reaction is reversible, it means it can go back. Citrate is converted to isocitrate, right? It's converted to isocitrate via our aconitase enzyme, right? So, I mean, let's just go and abbreviate the enzyme. So this is going to be our aconitase enzyme, all right? I'll just say ACO, just to make this short because we're trying to focus on the byproducts here. Now, when we go from citrate to isocitrate in this reversible reaction, which means it can also go back, pretty much isocitrate has the same number of molecules, the same number of atoms as citrate, but it's just a different structure. All right, so that's that. Now, let's talk about what happens from isocitrate to alpha ketoglutarate. And you know what? Just to make things less confusing, like for us, I'll try to mention all the small things that we talked about in the first video, like CO2 leaving and water being added and a coenzyme A group being added and things like that. But if I forget, then make sure to go check out my other two videos on the Krebs cycle where I actually cover the small things also. All right, so let's keep on going. So isocitrate is then converted to alpha ketoglutarate, and then so when it's converted to alpha ketoglutarate, we use isocitrate dehydrogenase enzyme. All right, so let's go and write that. So we use the enzyme isocitrate dehydrogenase. All right, isocitrate dehydrogenase enzyme. Remember, we went over the enzymes of the Krebs cycle in our previous video. All right, so if maybe these aren't as familiar, then make sure to refresh yourself. All right. So isocitrate dehydrogenase enzyme will convert isocitrate to alpha ketoglutarate. But we see the word dehydrogenase here in this enzyme means we produce some type of byproduct. Either it's probably going to be NADH or FADH2. Well, in this case, we're actually producing an NADH molecule. All right, so we're going to actually have an NAD plus molecule that's actually reduced, that gains hydrides or electrons and gets produced to NADH. All right, so that's this step, isocitrate to alpha ketoglutarate. And in this step, one thing, since I remember right now, let's go and write it down before we forget, is that we lose a carbon dioxide molecule, because what happens is that isocitrate is a 6-carbon molecule, and then we go to a 5-carbon molecule. So we have to lose that carbon somehow, and it's through carbon dioxide, all right? So next, let's keep on going. From alpha ketoglutarate, we turn into succinyl coenzyme A. So let's just go and write that down. Succinyl coenzyme A. And this enzyme, guys, is going to be alpha ketoglutarate dehydrogenase. So let's go and write that down. This enzyme is going to be alpha ketoglutarate. So alpha, should just draw on the alpha sign, alpha ketoglutarate dehydrogenase. All right, so, and since we see a dehydrogenase word here, we know that we're producing NADH or FADH2, right? Or either one of those two in general. So, but in this case, we're producing another NADH. So what's going to happen is that we have an NAD plus molecule gaining electrons, getting reduced, and we're forming an NADH molecule. All right, in this step from alpha ketoglutarate to succinyl coenzyme A. And since I remember again, let's go and write it down before we forget. Alpha ketoglutarate is a 5-carbon molecule, succinyl coenzyme A is a 4-carbon molecule. Where did that carbon go? Through CO2. All right, so you can see cellular respiration has a lot of waste. So our cells are constantly making CO2, which is why we have to constantly keep on breathing out our CO2 so our blood doesn't get too acidic and our pH doesn't drop. All right, so that we release the CO2 there. And one thing that I also want to mention is that we noticed that we went from alpha ketoglutarate to succinyl coenzyme A. So we added a coenzyme A group here. So let's go and write that down, all right? So we added a coenzyme A group. See, how do I do this? Let's just go from here because space is getting kind of tight, right? So let's go right here and let's say that we added a coenzyme A group here into this reaction, all right? And then from succinyl coenzyme A, what happens is that we actually go to succinate. So notice that the coenzyme A isn't there anymore. So we lose that coenzyme A group. All right, so we lose that coenzyme A group right here. And so let's go and write that down. We lose that coenzyme A group. And when we go from succinyl coenzyme A to succinate, if you guys remember from our enzymes of the Krebs cycle video, we use succinyl coenzyme A. I'm just going to write this out. Succinyl coenzyme A synthetase. All right, succinyl coenzyme A synthetase, which is catalyzing this reaction. So when we go from succinyl coenzyme A to succinate, we're still at a 4-carbon molecule here. We don't release any more CO2, right? But in this step, we just release a coenzyme A group. But what also happens is we see substrate level phosphorylation in this step. All right, so what do I mean by that? So when we release our coenzyme A group here from succinyl coenzyme A going to succinate, then what we see is that we convert GDP into GTP, right? So let me go and write this down. So in this reaction, let me just go and indicate that we're still in, as this reaction goes forward from succinyl coenzyme A to succinate, we have an inorganic phosphate that combines with GDP, all right, that turns into GTP, right? And then this GTP, since it has 3 phosphates, an ADP, an adenosine diphosphate, comes in here, right? An adenosine diphosphate comes in here, snags one of these phosphates from GTP, and turns into adenosine triphosphate, right? So we just made some energy here, right? We just made some ATP, and once the ADP snags a phosphate from the GTP, that means the GTP again loses a phosphate, so it goes back to GDP, right? So let's go and draw that out. So it goes back to GDP, and this is kind of like a little mini cycle. All right, so that's substrate level phosphorylation right here. All right, so this right here, everything that happens here is substrate level phosphorylation. Substrate level, oops, sorry about that. So in substrate level phosphorylation, just know that we make just a small amount of ATP, all right, not compared to oxidative phosphorylation like we'll see in the electron transport chain. So substrate level phosphorylation. Phosphorylation, there you go. So that happens in the step between succinyl coenzyme A to succinate. The second coenzyme A is released, all right? So that's done. Now we go from succinate to fumarate. All right, so succinate to fumarate. Now, this is the little exception that we were talking about when we were talking about everything happening in the mitochondrial matrix. This enzyme that converts succinate to fumarate is actually going to be bound on the cristae, and it's also going to be used in the electron transport chain as well. So this enzyme is going to be called succinate dehydrogenase. All right, succinate dehydrogenase. And succinate dehydrogenase is going to be the one to stimulate this catalyzation reaction between succinate to fumarate. All right, and when we go from succinate to fumarate, we don't really see anything that's happening other than making FADH2. That's the only byproduct that's made, you could say. So we just see an FAD, which is really important, by the way, because FADH2 and NADH are both electron carriers for the electron transport chain. That's how we get our energy. So we don't see anything happen here other than our FADH2 being made. So what happens is, let's go and write this out. We have an FAD molecule right here that actually gets reduced and turned into FADH2. All right, and so that's awesome, right? So we go from FAD to FADH2. So we make an FADH2 molecule in this step from succinate to fumarate, thanks to our succinate dehydrogenase enzyme. Now, then the next step, we go from fumarate to malate, right? Fumarate to malate via our fumarase enzyme. So let's go and write that down. So via our fumarase enzyme. And remember here, let's not forget that we add a water molecule here. Fumarase helps add a water molecule in this catalyzation reaction. All right. So we add a water molecule here, and then from malate, we go to oxaloacetate. Right? We go to oxaloacetate, and then from oxaloacetate we go back into citrate, but not, remember, we have to combine an acetyl coenzyme A group. So an acetyl coenzyme A group that's coming from the transition phase of glycolysis, make sure to go check that video out too, comes in, joins with oxaloacetate, making citrate. And that's pretty much it. And right here, we forgot to mention one thing, is that, wait, this doesn't even look like, this looks like a K, sorry about that. One thing we forgot to mention is that when malate goes to oxaloacetate, the enzyme that's used here is going to be called malate dehydrogenase. I don't know why I drew an arrow, sorry about that. Malate dehydrogenase. And our malate dehydrogenase enzyme helps create another NADH molecule in this step, all right? So we create another NADH molecule. So right here, let me go ahead and just draw it small, squeeze it in here. We get reduced. An NAD plus molecule gets reduced and forms an NADH molecule in this step from the conversion of malate to oxaloacetate. And then from oxaloacetate, we combine acetyl coenzyme A and make citrate through the enzyme called citrate synthase. All right, so we synthesize citrate in this step. So this enzyme that catalyzes the reaction between oxaloacetate combining with acetyl coenzyme A and making citrate is going to be called citrate synthase. All right, we went over the enzymes in the previous enzyme video, so make sure to go check that out. So that's it. That's pretty much what happened. That's the main byproducts that are produced in the Krebs cycle. And one thing before we end this video, is that all these byproducts that are produced, we said, was just from one acetyl coenzyme A molecule. But remember, if you go and watch that video, we said that we make 2 acetyl coenzyme A molecules in the transition phase of glycolysis. So that means that for each acetyl coenzyme A molecule, our Krebs cycle will turn once. But we made 2 acetyl coenzyme A molecules, so that means our Krebs cycle will turn twice. So let's go and label that. We're actually bringing in 2 acetyl coenzyme A molecules. So that means we're not going to make 1 NADH, we're going to make 2 NADHs. Right? We're not going to make 1 NADH here, we're going to make, right here, we're going to make 2 NADHs. We're not going to make, or right here even, we're not going to release 1 carbon dioxide molecule, we're going to release 2 carbon dioxide molecules. Right? We're not going to release 1 carbon dioxide molecule, we're going to release 2 carbon dioxide molecules. Again, we're not going to add in 1 coenzyme A group, we're going to add in 2 coenzyme A groups, right, in total, because our Krebs cycle is turning twice, one for each acetyl coenzyme A molecule. We're not going to release 1 coenzyme A molecule, we're going to release 2 coenzyme A molecules. right? Let's keep on doing this. We're not going to make, right here, we're not going to make 1 ATP, we're going to make 2 ATP. We're not going to release, we're not going to put in 1 H2O right here. We're going to put in 2 H2O molecules, 2 water molecules. We're not going to make 1 FADH2, we're going to make 2 FADH2s, right? And again, let's keep on going. We're not going to make 1 NADH right here. We're going to make 2 NADHs right here. And that's it, I think, right? I think hopefully we didn't forget anything. But so at the end, what the end goal of Krebs cycle is to make our electron carriers. And so let's see how many NADHs we made. We made 2 NADHs here, 2 NADHs here, 2 NADHs here. So that's 6 NADHs. All right, so let's go and write that down. What we made. So 6 NADHs in the Krebs cycle. All right, so 6. Let me just go and write apostrophe S right there. 6 NADHs. Let's go and see how many FADH2s we made. All right, and just remember this reaction when it goes from NAD plus to NADH, like in these, or even the FAD to FADH2. I just said 2 NADHs, but we're actually going from 2 NAD pluses to 2 NADHs. All right, so everywhere here. Same thing here with the FADH2s and right here with the 2 NAD pluses. All right, so let's just go and count how many FADH2s we made. So we just made 2 right here. All right, so we made 2 FADH2s. So let's go and write that down. We made 2 FADH2s, and let's go and see how many carbon dioxides we released. So we released 2 here and 2 here. So we released 4 carbon dioxide molecules in the Krebs cycle. All right, so let's go and write that down. So we release 4 carbon dioxide molecules in the Krebs cycle. Oh, right here, this is really important. We make 2 ATP molecules via our substrate level phosphorylation in the Krebs cycle. So we make some energy here also. All right. Okay, so let's go and write that down also. So we make some ATP. Okay, so we make 2 ATP molecules also in the Krebs cycle. All right, so let's see if we're forgetting anything else. I guess we know that here we're adding in 2 coenzyme A molecules and we're releasing these 2 coenzyme A molecules, giving us some energy to do this substrate phosphorylation, substrate level phosphorylation, right? Oh yeah, right here, we talked about these. These are just small things that we add 2 water here, 2 water molecules. And one thing I just noticed that we should state is that everything so when we go from isocitrate to alpha ketoglutarate, and then from alpha ketoglutarate, when we go to succinyl coenzyme A, all the catalyzation reactions starting from succinyl coenzyme A and on till oxaloacetate is all going to be reversible. It means that enzyme that can catalyze the reaction to go backwards too. So like this reaction right here, reversible. This reaction, reversible. This reaction, reversible. And then malate to oxaloacetate, reversible. But once it gets from oxaloacetate to combines with acetyl coenzyme A to form citrate, it's not reversible. All right, and then it starts reversible here, not reversible, not reversible, 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 and you get the idea, all right? So that covers it all. These are the main byproducts you can say that are produced right here, all right? And these are really important to know, especially these electron carriers that we're going to see play a huge role in the electron transport chain. All right, so I hope to see you guys in my future videos. I really hope this video made sense to you guys. In my future videos, I'll talk about more cool topics about biochem, physics, and chem, right? And maybe other topics too. And so I really hope to see you guys there. Don't forget to hit that notification bell, like, and subscribe, and see you guys later, and stay safe.